So, as some of you know, I'm at a bit of a transition in my career. I'm moving from Bangor via Helsinki to Aarhus on the 1st of January. And because I'm at a transition of my career, I thought, well, I didn't quite know how to do this talk. So actually, it's more of an aspirational talk than actually showing you past data. When I said that to my wife, she said, that's a cop-out, David. But anyway, there you go. So let's go. Okay, so I think one of the things is we really have to define the operational scales that we think are important, either on the micro scale over there or into the large scale. I put the smiley face in because I was too lazy to actually get an Arctic picture of ice extent. But it is very important that we actually understand really what we're trying to answer. And the key questions for me is, or if somebody said, well, what is the main question facing sea ice biologists, is how much biology is in the ice? I think we still don't know. And why, if at all, this biology is important on an ocean scale. And if it's important, why is it important? Is it important for the seasonal plankton dynamics? We've seen some talks already talking about that this morning. Is it important as a food source? Is it important in biogeochemical cycles in ice and the related surface waters? And is it important for this cryopelagic sediment coupling? Now, these are all the issues that people have talked about already this morning, so I guess it is important. And increasingly, this is what we're being asked to address, that if it is important, and I guess we know it is, how is it affected by a changing climate ocean? And a lot of the work that I'm going to describe is put into the context of this changing Arctic Ocean, although sometimes I think it doesn't need to be the only motivation for the work we do. So large-scale determination, estimation of what there is, we know that traditional coring mechanisms really are inadequate. You take a core 20 centimeters apart from it, you get one color hat, you take it another, you get another color hat, and sometimes there's nothing in it at all. So there is no remote sensing that is possible to look through the ice to see what biology is there. So with a group in St Andrews, we are developing some sensors to go on AUVs as well as ROVs, where you shine lasers of different light up to the ice and you measure the reflectance back. And as a result of the reflectance that you get back, you get a chlorophyll distribution. And I think this is one of the things that I think we can do a lot within ASP to actually work on. The alternative, of course, is to relate under ice light field to the ice biology and ice thickness. The problem is, as this picture really quite nicely shows, is the heterogeneity of a light field, especially when ice is breaking up, is a huge issue for actually deriving chlorophyll from under ice light fields. And I think that's why we're going the other way a little bit. The two have to be done together. Why is it important in a seasonal dynamics? Where you see from these graphs here is the blue is the ice, the under ice, and then the dark green is the spring bloom. The ice really does dictate where that spring bloom happens, and also how much food there is when there is no spring bloom just before it. And are we going to see a changing in these dynamics? Are we actually seeing, if you look at this rather than latitude, you actually look at it in climate change dynamics, are you going to be seeing increases in the spring bloom? The question that was asked by Dave, are you going to be seeing less or more ice algal production? Well, of course we don't know, but this is the kind of questions that I think we are expected to be able to answer and should be able to, because of course the whole of the ecosystem afterwards depends on that. The dynamics at the base of the phytoplankton spring bloom ice dynamics is key to basically everything else, as has been very elegantly said earlier this morning. So the quantity and the nature of the flux to the seafloor is obviously mediated by this relationship between sea ice and open water primary production. And of course this is our link to the past. And Eric's talk just now showed it in limbic systems. The chemical signatures to the past is what we need to be kind of studying a little bit more as well. Now the small scale is not uninteresting. A lot of people get into sea ice research because of the attractiveness of working at the micro level of the populations that are in the sea ice environment, and it's not uninteresting, as I've kind of been indicating up to now. But the ice is certainly hostile, but we need to put this word extreme into some kind of context. You know, this is the view from my office in Helsinki, and in the summer the plants and the animals are there at +20, and in the summer they're kind of surviving at -20. Okay, a lot of them are in dormant stages and sea stages, but in the ice is really only really productive at -2 to about 5 degrees. So, on the microscale, the changes in the spatial temporal habitats are the fundamental thing, and that's kind of governed by the physics. On the left-hand side, it's warm, porous ice. Sorry, on the other way around, and on the other side is cold ice. So the microstructure is the physics, and that's basically why we have to operate always with physicists. Now, slowly, this is from the Baltic Sea ice, we kind of are beginning to get some idea of the food webs within the ice and the relevance of DOM as being central to many of those dissolved organic matter, to the community dynamics within the ice. And one of the things that I particularly want to follow up is the role of DOM in the ice. And this follows on the work from Jodi's group in the back. They looked at a particular kind of DOM, EPS, and there's this kind of concept that the DOM is actually changing the physical properties of the ice, but it's also an important biological buffer against temperature, salinity, grazing, chemical stress, and biofilm. So I think one of the things that we're really going to see in the microstructure of ice is this kind of work. And again, this is from Christopher Krems, who came out of Jodi's work, this idea of the DOM continuum within the ice being related to the continuum in the marine system. Now, the two are not the same. The ice is a very much more concentrated microcosm, and it is wrong to take the principles we know from the open ocean and try and say that they're going on in the ice itself. So what do we need? Well, we really are urgently in need of long-term whole seasons, that's by which I mean water to ice and back to water again kind of studies, so that we can actually understand the physical, ecological, biological, molecular, and then the geochemical studies that come out of all of that. So we really are in an urgent need for these kind of studies to take place. And when they do, we do need them to take place over a range of oceanic systems. So coastal waters have a very different ice dynamics and productivity compared to the open ocean, compared to the very rapidly developing polynya systems as well. So we need to kind of establish, and I guess that's one of the things we're doing here, these kind of different ranges. And I know some people don't like the modeling aspect of it, but it's absolutely crucial, I'm sure, that we need to provide data that is coupled with and relevant data for the people who are doing realistic ice ocean physical biogeochemical models. They are desperate to actually talk to us, to tell us the kind of data they need, and that we can come provide it for them. Thank you very much.